The overall goal of this procedure is to demonstrate the technique of RNA interference in ticks by injection. This is accomplished by first synthesizing double-stranded RNA of the gene or genes of interest. The RNA is then injected into ticks, after which they are held in a humidity chamber for 24 hours. After this holding period, the ticks are allowed to feed on an animal, such as a sheep. Finally, tick feeding parameters, such as weight, molting, and oviposition, and the expression of the target gene or genes of interest by real-time RT-PCR are determined. Ultimately, results can be obtained that demonstrate the effect of silencing targeted genes on tick biology by evaluation of tick biological parameters and gene expression by real-time RT-PCR.
Our group works with ticks, trying to characterize molecular events at the tick pathogen interface, to use this basic information to develop methods for control of tick infestation and the transmission of pathogens to humans and animals. RNA interference has emerged as an essential tool in this research because it is the only method available for genetic manipulation of ticks.
The function of many tick genes is unknown. RNA interference allows us to define the role of tick genes and gene expression in many aspects of tick biology, including mating, reproduction, feeding, digestion, salivary gland function, and the tick vector competency for the transmission of pathogens. Demonstrating the procedure will be the tick RNA interference team from our laboratory, Dr. Ed Bluen, graduate student Ann Busby, and myself.
To generate double-stranded RNA, first synthesize oligonucleotide primers containing T7 promoter sequences for in vitro transcription of RNA. For example, for Dermacentor variabilis subbolison, use the oligonucleotide primers D8AAT75 and D8DVT73 shown here. Amplify the target gene by RT-PCR using 10 picomoles of each oligonucleotide primer and 1 to 10 nanograms of tick total RNA. Next, purify the PCR product, then use 8 microliters, or approximately 100 nanograms, to synthesize double-stranded RNA. To prepare the ticks for injection, first wash the ticks in the following series of solutions: tap water, 3% hydrogen peroxide, distilled water twice, 70% ethanol, and 2 final washes with distilled water. Perform each wash in a 50 milliliter disposable centrifuge tube by shaking, then decanting the solution through a fine mesh wire screen. Blot the ticks dry on paper towels. Then, aliquot the ticks into groups of 20 to 50, depending upon the experiment. Place each group into a 1.25 ounce plastic cup with a tight-fitting lid, and label each cup with the experimental group number. Next, assemble an RNAi team consisting of 3 people that will carry out the injection process. The first person positions each tick on double sticky tape affixed to a 3x6 inch sheet of red dental wax. The second person injects the ticks, and the third monitors the ticks after injection, breathes carbon dioxide on the ticks to activate them, and counts and transfers the living ticks into cups labeled with the experimental group number. All team members must wear disposable gloves. To begin the tick injection process, capture a tick using Dumont fine forceps and place it ventral side up on double sticky tape affixed to the sheet of red dental wax. Closely position the ticks into groups of 5. Place a small strip of masking tape over the mouth parts of all 5 ticks in order to further restrain them. Leave most of the body exposed so that the injection process can be observed by the tick injector. To inject the tick, pierce a hole in the lower right quadrant of the ventral surface of the exoskeleton using a monoject insulin syringe fitted with a 29-gauge half-inch needle. Next, immediately inject the ticks with 0.2 to 0.5 microliters of double-stranded RNA solution using a custom-made Hamilton syringe and a 33-gauge 1-inch needle with a 45-degree beveled point. Place the needle well into the tick cavity to ensure the placement and retention of the double-stranded RNA. Even though fluid is released after injection, deep placement of the needle for injection will ensure that sufficient double-stranded RNA is deposited into the tick cavity to cause gene silencing. Care should be taken not to over-inject the ticks, which will cause loss of hemolymph and death of the tick. Immediately after injecting the ticks, pick them up from the double sticky tape with the fine forceps and place them in a plastic recovery container. The ticks will briefly remain inactive after injection, but should soon begin to crawl around the dish. Activate the ticks by breathing carbon dioxide onto them. Once the ticks are crawling and active, the injection wound will heal rapidly and they will most likely survive. Count the ticks in each experimental group and place each group in a labeled plastic cup with a tightly fitted lid. Replace any ticks that die in the current group before injecting the next experimental group. Clean the Hamilton syringe before injecting the next experimental group by filling and then expelling the syringe with fresh 3% hydrogen peroxide 15 times, followed by 15 washes with sterile water. Take care not to bend the plunger of the Hamilton syringe, otherwise the plunger will not move smoothly and respond to the gentle touch required for tick injection. After the injecting, place the ticks in a chamber with a container filled with water and potassium sulfate, which maintains high humidity, and hold them for 1 day. Next, place individual ticks into tick-feeding cells glued to a sheep and allow them to feed with an equal number of uninjected male or female ticks, whichever sex was not injected. Collect and weigh female ticks from the feeding cell after they complete feeding for approximately 10 days, or when the control females have dropped off the host. Incubate each group of ticks in the humidity chamber until the completion of oviposition. Evaluate oviposition by group by weighing the egg's mass produced by all the ticks in a group. To evaluate the tick phenotype, after feeding, determine the number of ticks that survived and calculate the tick weight, as well as the oviposition and the fertility of the eggs. Depending on the targeted gene and objectives of the study, other analyses may be performed. To confirm gene silencing by RT-PCR, after feeding, dissect the salivary glands and guts from individual ticks from the control-injected and double-stranded RNA injected groups, and then extract total RNA from the individual tissue samples. Analyze the target gene transcripts in individual tissues by real-time RT-PCR and normalize the RNA levels against Tick16S ribosomal RNA using the gene norm method. Run dissociation curves at the end of the reaction to ensure that only one amplicon is formed and that the amplicons consistently denature in the same temperature range for every sample. Compare the normalized CT values for mRNA levels between control-injected and double-stranded RNA-injected ticks using the student's t-test. The protocol described here has been used in our laboratory for RNAi in many different exotic tick species. The amount of double-stranded RNA injected into the ticks varies with the size of the tick. Larger tick species can accommodate a larger volume. The references can be found in the accompanying written portion of the protocol. If the protocol is done correctly, less than 5% mortality should be obtained from the injection procedure after 24 hours. A typical phenotype after gene knockdown in ticks is shown here, where a panel of ticks were injected with pools of double-stranded RNA that were used to screen for tick-protective antigens. In ticks that are examined by light microscopy, cellular degeneration in tick tissues may be observed. Phenotypic changes may also be accompanied by loss of function. For example, RNAi of subolacin results in sterile males that cannot mate successfully with the females.
Following RNA interference in ticks, other methods can be used to determine the impact of gene silencing on gene and protein expression and on tick biology, including real-time RT-PCR, light, electron, or confocal microscopy, genomics, or proteomics. RNA interference can also be done in tick cell cultures and provides an in vitro method for the study of tick-cell pathogen interactions.